In this video, we will discuss some of the bioinformatic results that further validates the relationship between KRAS and FAS and the CD8 plus T cells, FAS receptors on the immune cells. So first they check the single cell RNA-seq data and find out what are the different kind of T cells population using the Disney plot, and simultaneously they also check what are the different kind of pancreatic cells, that is ductal cells type 1 and type 2 they see in the single cell RNA-seq data. And from the clusters that they obtained about the T cells, uh, they distinguished those T cells based on the transcript for different kind of T cells that are present in the single cell RNA-seq data, that is CD3d T positive T cells, CD4 plus T cells, CD8a T cells, FAS, and they also check this FASL ligand in those T cells. And as they have previously find out that CD8 plus T cells are the one that expressed most of the FASL ligand, they further validated this they using the single cell RNA-seq data of the naive PDAC tumors. And we can see the CD8 plus T cells expressions in the different metaclusters seems to be overlapped with this CD8 plus T cells expression in different metaclusters. There is some overlap in CD4 plus also, but that overlap is minimum, and the CD8 plus overlap is high with the FASL ligand compared to the CD4 plus T cells overlaps. And once validated that it's the CD8 plus T cells that has this FASL ligand expressions, they wanted to check the expressions of this KRAS and FAS in those single cell RNA-seq data. And what they have find out is that the overlap of KRAS and FAS is almost negligible, and they can see that in the panel E, the quantifications shows that the KRAS high expressing cells has this low FAS expressions, whereas KRAS low expressing cells has high FAS expressions. So this validates their relationship, that KRAS has inverse relationship with the FAS that they have find out from their previous experiments. Next, they check the cancer cell genome data and separate the populations based on these two categories, that is KRAS high cancer cells and the KRAS low cancer cells, and then check different immune cells that are present in these two populations. And what they have find out is that in the case of this KRAS low populations, they see the more CD8 plus expressions, and whereas in the case of this KRAS high populations cancer cells, they see the low CD8 plus expressions. And this is also in line with their finding that they observed in the previous experiments, that KRAS low cells has more CD8 plus infiltrations, and they can interact with the FAS receptors on the tumor cells and trigger apoptosis. So next, they check the survival rates of these two different kind of populations from the cancer genome atlas data, and they have find out that KRAS low populations have more survival percentage compared to the case of uh, KRAS high populations, KRAS high uh, data. So this all indicates that the KRAS is epigenetically KRAS modulates FAS receptors epigenetically, and the high KRAS tumor cells has low FAS receptors, and thereby the immune cells will not be able to interact with this FAS receptors on the tumor cells and not being able to trigger apoptosis. And vice versa is also visible in the KRAS low populations. So I hope you understood the relationship between KRAS mutations, FAS receptors, CD8 plus T cells. So in this panel, they check for KRAS, not only wild type KRAS but also the mutated KRAS. So I hope you understood the message of this paper. So if you need to understand more about these experiments or bioinformatics analysis in detail, you can go through the reference that has been provided in the description. So that will be the last uh, result experiments that we are discussing about this paper. If you want some more details, you can refer to the papers for like experiment supplemental results. That's all for this video. Thank you for your time.